Thank you. Welcome back to the final session of this meeting, which I've enjoyed, and I hope you've enjoyed it as much as me. So I'm going to introduce our last keynote lecturer, Mike Murphy, and he trained at Trinity College Dublin, and then he's had a long journey through science, which he took up apparently because he turned down or he didn't get the role of James Bond. Apparently, I don't know which James Bond he was following, and he now obviously is an investigator here in Cambridge at the mitochondrial biology unit, and has a huge track record of work, including developing MitoQ and other agents. And I won't steal his time, and I'm going to invite Mike to come up and give us his lecture on mitochondria and ischaemia reperfusion injury. Thank you.
Okay, thanks, Shamima. That's nearly all true. What I'm going to do is go through some of the background that we've been working on with colleagues in Cambridge and around the world on the field of ischemia reperfusion injury, how that relates to mitochondria, and how we've gone from basic science right the way through to a potential therapy that we're now trying to see if we can develop that. So we're taking it right the way through from the basic science. It's not directly related to primary mitochondrial disease, but it's a consequence of mitochondrial damage, as I hope to convince you. So there we go. Looking at what is ischemia reperfusion injury, most of you are familiar that it contributes to things like heart attack and stroke. And we know that you'd have normal oxygen flow into a tissue, you get a blood clot, and this will precipitate a heart attack or a stroke or something similar in other tissues. And then you want to repair this or remove the ischemia and restart the blood flow as quickly as possible. And that will be done in the heart by PPCI, just to get a stent in or remove the clot. And mechanical thrombectomy in stroke, or with clot-busting agents like tissue plasminogen activator. You restart the tissue flow, and you need to do that to recover, to stop the cells from dying. But ironically, during that reperfusion, you get an extra burst of ROS production. And it's that burst of ROS production that's called reperfusion injury, and we're interested in that because it was thought to be related to mitochondrial ROS production. So when we started this about a decade ago, the origins of that ROS production were unclear, and it was unclear if it was actually a random process or it was something that was druggable. So what we're doing is trying to understand what's going on reperfusion. The prime model that we use is the heart. That's a heart. And what happens in a heart attack would be that a blood clot would block a coronary artery. The area downstream would become ischemic and would start the cells would start to die. And you have to get in as quickly as possible to the cath lab in the hospital, put in a stent or to remove the clot and reperfuse that. And so the model of that reperfusion injuries thing we're trying to address is a mouse model that we use, the LAD mouse model here. You can open up a mouse, you can tie off a coronary artery, the area downstream would become ischemic, mimicking what would happen if you have a heart attack, and then you can untie this and reperfuse and mimic the reperfusion injury. So it's a very good model of testing drugs and the mechanistic aspects of reperfusion injury. In addition, you can sew up the mouse again and you can test the long-term damage months afterwards to see if your therapy has had an effect at that stage. So, what happens when we do this? Well, what we did is trying to understand what happens in ischemia. And what we found is in the ischemic zone of tissues, you get a huge buildup of the mitochondrial metabolite succinate. We can illustrate this very nicely here. And this is what's called mass spec imaging of a heart. You freeze the heart, take slices through, raster across with a beam, and you can see in the area that became ischemic, you've got this huge buildup of the metabolite succinate you're measuring by mass spectrometry, and you can see that it's got a huge buildup in the ischemic zone. So what's happening during ischemia, the first thing we discovered was that during ischemia, you get this dramatic accumulation of succinate. What happens is that cells are set up to accumulate succinate under certain conditions, such as glutaminolysis in inflammation, but this gets inadvertently switched on during ischemia. What happens is that you get breakdown of purine nucleotides, quite dramatic breakdown of purine nucleotides during ischemia. This leads to metabolites which end up in malate outside here, and the malate gets exchanged for succinate and goes back into succinate here, and then you get this huge accumulation of the succinate building up outside and inside the mitochondria. And this can be up to 10 to 20-fold increase in succinate. It's got a dramatic effect. So that's what's happening during ischemia, but how does that contribute to this putative ROS burst upon reperfusion? Well, what we hypothesized was that succinate's kind of storing electrons. It's storing electrons that build up during ischemia, and then as soon as the oxygen comes back in, it's dumping the electrons on oxygen to produce ROS. Illustrate this here. If we repeat that experiment but slice through the heart after reperfusion, you can see the succinate's all gone. We can do this a bit more quantitatively by taking sections of the heart and just measuring the succinate by quantifying the succinate using LCMS on the tissues. And what you can see here is that you get this dramatic buildup of succinate in the tissue, and it goes back to baseline within a couple of minutes. Same in the in vivo heart, back to baseline within a few minutes. So the succinate's been accumulating, and then it's oxidizing very quickly. So what this is suggesting then is that we're actually getting somehow the succinate is dumping the electrons it's storing onto oxygen to form ROS. It had been known for a very long time back in the history of mitochondria, before all you were born, that you had, nearly all of you, that what happened is that succinate was actually a very good way of making mitochondria produce reactive oxygen species. What happens is this process called RET, or reverse electron transport, happens. If you give succinate to mitochondria, it gets very rapidly oxidized by succinate dehydrogenase, reducing the Q pool. A bunch of the electrons go downstream, pumping out protons, building up a large protomotive force. But because complex 1 is close to equilibrium in many situations, this is sufficient to drive electrons backwards from the Q site through the iron sulfurs onto the flavin mononucleotide, we think, where it produces superoxide. And this was known for a long time. It was thought to be a lab curiosity. To illustrate it here, what you can see is if we take isolated mitochondria, we feed them with succinate and we measure where the superoxide goes. It will go to hydrogen peroxide, it will diffuse out of the mitochondria. We can measure the hydrogen peroxide coming out of the mitochondria, and you can see buckets of hydrogen peroxide coming out, illustrating superoxide production by RET. Far more than if you abuse your mitochondria with antimycin, and if you just uncouple them with a bit of FCCP, you abolish this production. Take-home message, mitochondria are designed to produce reactive oxygen species as a signal under certain circumstances, not just when they've been abused or you've treated them harshly with something nasty. That we think is a signal in things like inflammation, but it's been activated, we think, inadvertently upon reperfusion. So, can we measure this? Well, we go back to our LAD model, and we want to measure ROS production in vivo. Measuring ROS in vivo is very, very challenging because the fluorescent methods can't really work in vivo. So what we've developed over the years are mass spec methods where you can measure ROS production inside the animal, because you have to do it in situ, where the calcium, oxygen, ATP, and so on levels are identical. You can't extrapolate ex vivo. You can't measure the ROS ex vivo. You can't interpret what's going on in vivo from isolated mitochondria. It's just too complicated in there. So what we do is we use this targeting group that we've used for a long time, the triphenylphosphonium, that has the trick that it will go straight through membranes and accumulate in mitochondria in vivo several thousandfold. We've used that for delivering lots of stuff to mitochondria. We do this one we call Mito-B, because we call everything we make Mito, and B because it has a boron atom here. Goes into all the tissues, goes into the heart, for example, accumulates there, and hydrogen peroxide reacts slowly and selectively at a very slow rate with this to form a phenol. And this gives you a chemical signature of hydrogen peroxide production. And because they're both accumulated to the same sort of extent, the ratio, which we can measure by mass spec, by LCMS relative to internal standards, will give us a rough indication of how much hydrogen peroxide is forming inside there. So the ratio is low, not much. If it's high, lots. So we can do that. We inject that into the mice. We take out the tissue that was exposed to reperfusion when we reperfuse it, and we try and see, can we measure ROS production? So there you go. This ratio is low in the control. After reperfusion, it goes up. So that's consistent with a ROS burst upon reperfusion, very early upon reperfusion. However, that could be by many different sources. So we're not sure if that illustrates that it's being driven by the succinate. To test that, what we do next is just to inhibit succinate accumulation and oxidation using an inhibitor, which is just malonate. Now, obviously, we knew that malonate couldn't possibly get into cells, so we used an ester as a prodrug. And you can see here dimethyl malonate is getting into the tissue. If we administer that before the ischemia or the reperfusion, gets in internal esterases, provides with malonate, and we block succinate dehydrogenase, which will stop the accumulation and oxidation of succinate. When we do that and repeat the experiment, you can see that here the mito-P to mito-B ratio has gone right down. So again, we've blocked this ROS production by blocking succinate. So that suggests that succinate accumulation and oxidation is driving this ROS production. Corollary of that is that if the ROS production is driving the reperfusion injury, then what we would expect to see is that there should be less damage to the tissue. We can measure that histologically, and what you can see here, we take slices through the heart. The untreated heart, the white area is dead, the red area is alive, and you can see this white area seems to go down a bit, and you can see that the area was ischemic, about 40% has died in the control, far less with dimethylmalonate. So that's consistent with the idea that we are actually getting ROS production early on reperfusion driven by the succinate oxidation. The next point then is, well, we have assumed that it's by this RET process here, okay? But that's quite difficult to test in vivo. It's been tested a few ways by us and by others. The first thing you can do is you can add uncouplers, and that will prevent this increase in protomotor force, and that blocks this ROS burst upon reperfusion. You can also add inhibitors of complex 1, such as rotenone, that will also block this ROS burst upon reperfusion. So all of those would point to this mechanism of ROS production being driven by RET at complex 1. However, we'd like a more direct test of this, and very luckily, we have this mouse model that Doug Wallace had in Philadelphia, where there's ND6, in the ND6 gene in mitochondrial DNA, has a single nucleotide substitution, given a single amino acid substitution in the heart of the ND6 subunit within complex 1. And what Doug and his colleagues suggested was that complex 1 with this mutation was not able to undergo RET. So we tried to see if we could confirm this when he very kindly gave us the mice. And here you can see heart mitochondria. If you give them succinate control, heart mitochondria produce buckets of hydrogen peroxide by RET. The ND6 ones don't, so they're not producing ROS by RET. If we then measure mito-P, mito-B ratio in those mice, you can see the control mice here. We get an increase with reperfusion, but we don't get an increase in the ND6 mice here. So again, that suggests that the RET is essential for the ROS production. However, again, a corollary with that should be that we should be able to see a protection against the tissue damage here. So if we look at this, again, you can see the wild type. The white area shows the dead bits, less white in the ND6, and we can quantify it here. So the ND6 mice here seem to have far less damage to those tissues there. So what we have then is an indication that we have a system of ROS production that goes from succinate through to the Q-pool, high protomotor force, and complex 1 drives this burst of ROS as it uses up the succinate upon reperfusion. But a corollary of that then is, if you've figured out the mechanism, if it is going to be RET, we need not just a reduced Q-pool, we need a very high protomotor force. And what we're saying then is that as soon as we reperfuse the heart or whatever tissue, the protomotor force has to go back up to a very high level in the first minute or two of reperfusion. Otherwise, this mechanism can't work. We need that high protomotor force, high membrane potential. So how could we measure that in mitochondria within a heart? Very difficult to do. So this is where I started working with Bob Balaban and a very good PhD student, Abigail Giles. So for her PhD was joint with Bob Balaban and the NIH and back and forth to Cambridge. And they were working on the stuff that Bob has been working on over many years, which was to try and measure optical changes within the isolated perfused heart. But what are we going to use in the heart to measure membrane potential? Well, we can't use dyes for fluorescence, or we can't use lipophilic cations because the timescale of changes is not going to be accessible. We need something endogenous. So what we're going to do is use an endogenous membrane potential monitor, which is cytochrome BC1 complex. The bit of that can monitor the membrane potential are the two B hemes, the low potential and the high potential. So you remember at your mother's knee when you learned about the Q cycle, you remember that the B hemes, the electrons go back and forth between the two B hemes as part of the way of storing the reduction potential going from Q to C as a membrane potential. So what happens is there's a single electron distributed between the low and the high potential heme here, and they're interacting with the membrane potential here. And so at high membrane potential, the electrons are kind of pushed over here. At low potential, they're pushed back here. So the distribution of the single electron between the low and the high potential heme gives you some indication of the membrane potential here. But of course, we need to measure that, but luckily, these things all absorb in the visible, hence cytochromes, and we can follow the changes in absorption of this, which indicate whether it's reduced or oxidized. So we can measure the distribution of that from the spectra of BL and BH. Now, to do that, though, is a bit more challenging. So this is where Bob and Abigail came in, and what they're able to do is to do spectroscopy on the isolated intact heart. What you do is you take a Langendorf heart here, you put a light catheter inside the left ventricle, and then you have light shining through. So it's just like you're shining light through a sample. You're going to take a spectrum. To actually capture all the light, because to deal with all the scattering and the changes in thickness, you have to put the heart inside a chamber like this where we capture all of the light. It gets reflected down to a light guide, and then we get the spectrum here. So what we do with that is we have now a spectrum of the absorbance of the heart here. The next bit is to say, well, what do we do with that spectrum? Here's what the raw spectrum would look like when you deal with a whole bunch of other changes of background scattering, optical effects of the machine. What you do with this is you say, that's my experimental spectrum. And what I do then is we have these known spectrum of reduced hemes and the other cytochromes absorbing in this region here. And these are the idealized spectrum. What you do is you do a linear superposition of these spectra until you get the best fit. And so we put those together in certain ratios, and we'll get something that seems to mimic the spectrum there. And then to know whether we've got the right spectrum, we subtract the best fit from this, and we get a residual, which shows that this is the absorbance of those hemes inside the heart at that moment. So from that absorbance, though, we need to try and see how do we get the absorbance of the BH and the BL and get that into something that we can measure as a membrane potential. Also, the complication is that there's a single electron on the BC1 complex, or there's no electron. We only want to look at the BC1 complex with a single electron. And so what we do is we go from the absorbance of the low and the absorbance of the low and the high. We sum this, which gives an indication of how much, of what proportion of the complex has an electron. And then this ratio to those, we call FBL, gives you some indication of the membrane potential. As you can see here, this parameter is high when you have a lot of membrane potential with succinate and nigericin. It goes down with malonate, with ADP, and with uncoupler. So you've got something that's responsive to membrane potential, this parameter here. But how are we going to get from that to the membrane potential? Well, here what we do is we go back and do an orthogonal measure of membrane potential where we measure the B hemes and also the membrane potential by uptake of TPMP, a standard way of measuring membrane potential in isolated mitochondria. So here we measure membrane potential by TPMP accumulation within the chamber, and simultaneously we measure light. And you can see that under a whole bunch of different conditions, you get this shape here. And you can see you're getting a very good dependence then of this parameter, which we can measure in the heart against this calibration. And so if we measure FBL in the heart, we can read off a membrane potential inside the mitochondria within the heart. So when we do that, we try and go back to, by now you've probably forgotten what the point of this was. The point was that we thought that the membrane potential must go up very quickly upon reperfusion. And so now we go back to the heart, use this method and see, can we measure the membrane potential upon reperfusion? So the heart's back in there, and we have equilibrate for a wee bit, ischemic for a bit, and then we reperfuse. And we can measure the spectrum every second, and so we've got to measure the mitochondrial membrane potential within that heart every second. So this is the output of the membrane potential that you can see. So here's the membrane potential inside the heart during normoxia. As soon as it hits ischemia, it goes right down as you might expect. There's no oxygen. We think here what's happening is you still have a very high membrane potential, and this is being supported by ATP production from glycogenolysis to glycolysis, and that ATP is then going into mitochondria and reversing the ATP synthase. So we think this here is indicative of sustaining the membrane potential by ATP hydrolysis during ischemia. At this stage here, you get a precipitate drop in the membrane potential at this point here. What's happening there is that the ATP is all being used up by hydrolysis through the mitochondria, and it's also coinciding with what's called hypercontraction. There's not enough ATP and ADP around for the actomyosin filaments to separate, so you get hypercontracture, change in cell wall. Then we reperfuse, and what you can see here, gratifyingly, the membrane potential goes shooting up. There's a bunch of other changes coming along later, which will be due to maybe permeability transition pore and damage to the mitochondria possibly. But it's this bit here we're most interested in because it's going back up to baseline levels very quickly. If we expand that area, what you can see is that as soon as we reperfuse, the membrane potential does seem to re-establish itself within less than a minute. So that's gratifying. So as a minute, you can see we've actually got this re-establishment of the membrane potential. That's consistent with our model then, that even though the heart has been abused and left at a very low potential, as soon as the oxygen comes back in, it'll oxidize the succinate that's been accumulated and drive proton pumping and drive RET and high membrane potential. We can show that's due to succinate because here we measure also the levels of succinate in the tissue, and what you can see in the whole heart, the succinate's here, and you can see upon reperfusion it goes down to baseline here, and so this suggests to us that the succinate oxidation is driving this repolarization of the mitochondria. Then later on other things will start to come in and the membrane potential will change, but this is the section that we're most interested in. So what we think then is that we have a complete explanation for ischemia reperfusion injury. We're trying to convince the field or wait for people who don't agree with us to die. But that's the model that we're trying to put out there, that we get the succinate accumulation during ischemia, oxidation upon reperfusion, and then we get this burst of ROS production, regeneration of the membrane potential, and the ROS initiates cell death. We think the ROS then causes damage, releases DAMP, such as oxidized mitochondrial DNA, oxidized cardiolipin. It also induces the permeability transition pore, induces cell death. And of course, that will change a little bit within the lesion. It'll be different types of cell death at different parts of the lesion. We think that initiates a lot of the damage. Then that leads on to, and we think this occurs in the first minute or two, that then leads on to downstream damage, downstream cell death that will lead to the scarring and changes to the heart. If that's the case, we've learnt a couple of things. The first is the ROS burst occurs very early on in reperfusion injury. So it occurs over the first few seconds. That means your therapy has to be, if it's aimed at this pathway, it has to be there at reperfusion. It also explains why so many trials in ischemia reperfusion injury never worked. People were adding antioxidants an hour later, 10 minutes later, all those things. That's why we think targeting this process but at reperfusion should be the way forward. So, how can we use that knowledge to develop drugs? Well, we showed you earlier that we could block succinate accumulation oxidation with dimethylmalonate. So we think this has got, and we could show that that was protective. So that here we're using it as an indication of mechanism, but it also suggests that this process could be a drug that we could use then to potentially block ROS production at our injury. So we started exploring this. So bear in mind, how would this look in clinical practice? Not that I'm ever going to go near anything in clinical practice, I faint at the sight of blood, but for all you brave clinicians out there, this is probably what you would do if you had a heart attack. You get a heart attack, you get a pain in your chest, you'll end up in the cath lab, there'll be a catheter going right the way through into the coronary artery to pull out the clot and block the clot and restart perfusion. So what's going to happen then is that this is this timeline of treatment. So if you know you're going to have a heart attack, you can start loading up on Mitochondria at this stage here. So plan ahead. If you don't plan ahead, this is the only time that you can actually be treated with a drug or downstream to treat the other damage. So this is the time you've got to get in there to stop the reperfusion injury. You can start repairing the damage a bit later on, but to stop the damage, we would argue you have to be in here. When you're inserting the catheter and you're pulling out the clot, you need the drug at that stage there. So let's mimic that with dimethylmalonate. And what you can see here is in a mouse model, if you give the dimethylmalonate, the ester form here, prior to ischemia, it's protective. But if we give it here at reperfusion, just infuse it as we pull out as we untie the coronary artery, it's not protective at all. So what's going on here? Well, what we think is happening is the dimethylmalonate is getting in, probably, but it's not getting in quickly enough, and it's not getting broken down quickly enough. Because remember, it's got to be there, delivered, active, and the active drug has to be there in the first couple of minutes. So with Richard Hartley in Glasgow, we decided, well, let's make some esters that still will get into the cell, because obviously the issue was malonate won't get into the cell, so we needed to make something that was masked and would get in. So we made a whole bunch of these esters which are hydrophobic enough to get in, and they will be rapidly hydrolyzed. And this one was very effective as diacetomethoxy methylmalonate, a very standard approach for masking carboxylic acid groups. And in this situation, what we could find was that was a very effective drug. So we thought this is the one we might start taking forward to the clinic. But then there were reports that malonate itself was effective. Now this was impossible because remember I said malonate can't get into cells because it's well known that dicarboxylates are unable to get through the plasma membrane, unable to get into cells. So we tried to test that. So here's malonate. So here's our control experiment to show the other people were wrong. We give disodium malonate here. Doesn't be protected, but you give it a reperfusion. It's a far better drug than the other ones we spent a year making. So there we go. We now have a very good drug. We give it reperfusion. It's very effective, but it shouldn't work. So what's going on? How is it? We did a bunch of other stuff to show it was getting into cells. It was acting to block rust production and so on. So it was acting in the way that we anticipated we wanted things to act. But how is it getting in? Then here in Prague in my lab, and Thomas Krieg's lab, showed what is the mechanism of getting it into cells. We know that once malonate is inside the cell, it's very rapidly taken up by the dicarboxylate carrier and goes in to inhibit succinate dehydrogenase. But how's it getting in? Well, with a lot of work over a year or so, Hiran worked out the mechanism of uptake of malonate and why it was getting in and why it was protective. What's happening is illustrated here. We have a Langendorf heart, a perfused heart. And if we perfuse the heart with malonate and measure how much is getting into the tissue, none's getting in. But if we hold the heart ischemic for a few minutes and then perfuse, some gets in. And the longer we hold it ischemic and then start perfusing with malonate, the more that's getting in. So we're getting very large uptake of malonate only in the ischemic heart. And that uptake then, this has died, that uptake then is through a particular carrier, MCT1. And the MCT1, that is the monocarboxylate transporter. And what happens with that is that is the mechanism for malonate uptake, and we can block it with this particular inhibitor, which shows that it's very selectively been taken up by that carrier. But why is it getting in, and why is it only getting in during ischemia? Well, what happens during ischemia is the pH drops to about 6.5. Malonate, the pKa of malonate is about 5.8. So normally at pH 7.4 in the blood, it's all diionic, it's highly charged, won't get in. But at low pH, it's actually going to start to be protonated, the monocarboxylate. So it's going to be 10 to 50% in this form here, which is a monocarboxylate and is a very good substrate for MCT1 and the other members of the MCT family, and will exchange for the lactate that also builds up during ischemia. MCT1s are used for carrying lactate, and they're very highly expressed in all tissues. They carry lactate, but they also carry other things like ketone bodies, so they're quite promiscuous. They'll take quite a lot of different substrates, so this means that it's kind of the perfect carrier for this, because it will carry through the monocarboxylate form of malonate very nicely. So from that, what we do then is now we're developing, as the therapy is going to be malonate in a slightly acidic form. So we'll take the malonate and we'll make it acidified, and what you can see here is this is going to be our drug at about pH 6. We infuse that, but it has to be infused at reperfusion to go in through MCT and then do its job here, okay? And we've shown that's very effective in the heart, given at reperfusion. Also, if we look at those mice a month later, there's far less scarring, and echocardiograms and heart function are vastly protected by that. So we've got long-term protection in the heart. So that's good. But could we also extend this to stroke? Heart attacks are reasonably well looked after by these kind of coronary interventionists. What will happen if you get a heart attack? You've got a very good chance of surviving. You'll be given drugs like clopidogrel, aspirin, and so on. So very few people die during the procedure. What happens is they'll have heart failure over the subsequent years because you're still getting the reperfusion injury. We predict that if we gave malonate at reperfusion, we'd have far less heart failure later. But the interventionists, they're on the golf course by then. They're not worried about what happens later. Also, as a clinical trial, it's quite hard to design. But stroke is a real problem. Stroke is a huge unmet need. And but how could we treat something in stroke if we want to give the drug at reperfusion. Because the standard of care for stroke for a long time was things like tissue plasminogen activator. In other words, if you had an ischemic stroke caused by a blood clot, you would take these drugs which would activate the digestion of the clot and reperfuse. But that reperfusion would be relatively slow over a period of time. So in other words, giving malonate would not be so good because you wouldn't be sure when you should be given the malonate. But since then, we have this new intervention called mechanical thrombectomy, where we treat strokes in large blood vessels in the same way that we treat heart attacks. We put in a stent, we go up into the brain, we pull out the clot, and we reperfuse that part of the brain. So this means we know exactly when we're reperfusing. That opens up the possibility of taking malonate drug at the same time. Here are some of the pivotal studies that will lead to mechanical thrombectomy becoming the standard of care. And what you can see here, this is the old standard of care, say tissue plasminogen activator, and this is called a modified Rankin scale measured at 90 days. And what you can see is 5% of the control population, or the population having a stroke, come out with no problem. 6 is dead, so that's not good. And this is increasingly bad, so these are going to be bedridden. So everyone here, a huge proportion of the population having a stroke, are having a really bad outcome. Even with mechanical thrombectomy, it shifts it to the right, but we still have a lot of dead people and a lot of people whose lives have been ruined. So it's a huge unmet need. And this, we'd argue, a lot of this extra stuff here, part of that's being caused by the tissue damage because you're not getting the mechanical thrombectomy in quickly enough. But in addition, even when you get the mechanical thrombectomy in there at time, you're still getting the reperfusion injury. If we can put in a drug to stop the reperfusion injury as we reperfuse, then we have a better chance of pushing this to the right. So, Jordan Lee, a very talented PhD student who just completed his PhD a short while ago, set up a model of transient middle cerebral artery occlusion inside a mouse where we could actually do these experiments and deliver the drug locally, as we would hope to do in the patient. So what you do is you go into the mouse, you put in a little catheter, and you put a suture here which just blocks the middle cerebral artery. And here you can see the blood flow measured by Doppler or laser Doppler flow metry. And then you pull it out and you get reperfusion. So that's the idea. Easy said than done, but very, very technically challenging to do. And to show that during this model we were actually having the same procedure, what you can see here is the succinate levels inside the brain of the mouse. The brain studies are great because you can use the other hemisphere as a control. So here you can see on the top, this is the area that's ischemic, and reperfusion is hugely damaged. This is by superposition of many different measures to indicate the area damaged. But here's imaging mass spec, and again you can see this huge bright area of succinate accumulation which washes out on reperfusion. So the same mechanism we think is going on in the brain as in the heart. And this is what we would hope to do then in the patient. We'd put in a catheter, we'd have a microcatheter, deliver the drug post the clot, have the drug present here, and pull that out. And what Jordan was able to do is to set this up inside the mouse brain. We have the occlusion, we have the infusion catheter, we give the drug at the same time as we pull out this suture, and we can get reperfusion. Obviously, the blood-brain barrier is a huge issue with any drug going into the brain. We were optimistic because we know there's huge amounts of MCT 1, 2, and 4 in the brain in all the cell types there because there's huge amount of lactate transport and ketone body transport in the brain. So we were optimistic we need to demonstrate that for sure. And also hugely expressed in the endothelial cells that make up the blood-brain barrier. So here we just want to check, are we getting good delivery of malonate into the brain? So we're not looking at ischemia. This is just the malonate in the brain tissue. Do a tail vein injection, we get some in. Do a local injection, just into the cerebral, intra-arterial injection, we get some in. We do acidified disodium malonate, get far more in. And if we block the MCT with this drug here by pre-delivery, we block that uptake there. So in other words, we are getting good delivery, and it's getting into the tissue through this mechanism that we thought. But is it actually causing any protection? So here we do MRI at 24 hours after the stroke, and here you can see the white area here is where we're just putting saline in. Here, what you can see here, extensive brain damage, and you can measure that again by histology, but with acidified disodium malonate getting far less of that brain damage there. We can quantify that here, where here you can see this is the infarct volume, goes down, measured by MRI, and again measured by histology. And more importantly, we can also do this measure here, the Bedison scale, which gives you a rough indication of neurological outcomes. So the green show the ones treated with acidified sodium malonate, and they're all down to the lower number. Lower means better outcome, and this correlates with infarct volume measured either by MRI or by histology. The mechanism then, well, we could show that we were blocking oxidative stress inside the brain. In addition, if we go back to our ND6 mouse model, which remember the complex one does not have ROS production, and again what you can see here, in those mice with the ND6 mutation, we get far less damage to the brain measured here by histology. So again, this also tells us that about 50 to 60% rescuable tells us that that's the maximum we would get by blocking this process, and very gratifying. This number, say 55 to 65, is about the percentage we get with malonate, suggesting that we are blocking this type of cell death to the extent that we can with this drug. So, what we hope then is convincing you that we have a way of attacking stroke. So these were the data I showed you before, that we've moved from this to this, where we still have huge unmet need. There's huge human and medical and economic costs here. But if we can add mechanical thrombectomy and give acidified sodium malonate, which we can do using the same catheters that are currently used in stroke, if we can push these over to the right just a little bit, the benefits then for patients would be huge. So that's the take-home message. By understanding the mechanism of our injury, we've been able to leverage that knowledge, a few surprising twists and turns, and come up with a very simple drug that we think should be effective given at reperfusion for heart attack, for stroke, but also for many other things such as resuscitation of patients, organ transplantation, all of those areas where reperfusion injury is a major aspect of the damage. So this is the team. All this work is done with my close collaborator Thomas Krieg, who is a clinician and expert on reperfusion injury. The chemistry is done in Richard Hartley's lab. Hiran did a lot of the stuff here on developing malonate as a therapy, and Jordan did the stroke stuff there. Abigail, working in Bob's lab in Bethesda and in Cambridge, set up all the measurements of measuring membrane potential. So hopefully I'm in time, and hopefully that was of some interest, and now happy to take questions. Thank you very much.
Thank you, Mike. That was a great story. Do we have questions for Mike? And before Joe, can I ask a very quick question? Have you had access to cells from patients with MCT1 deficiency to test if that's actually where it's going in? I mean, I'm sure you're right, but I just thought it might. I know you can't get ischemic hearts from patients with MCT1 deficiency.
We've done it in MCT1 is essential. We've done it in a heterozygous MCT mouse.
There are patients with MCT1 deficiency.
Yeah, but it's to do a full knockout. Though.
Oh yeah. Now I was thinking about humans rather than mice.
Yeah, that's something we should look at. That would be interesting. Yeah.
Thanks, Joe.
Joe Polton-Oxley. Lovely, fantastic. And of course, a question that I expect everybody else knows the answer to is whether succinate goes up in stroke-like episodes, and also, would it work?
Don't know.
Do people know whether succinate goes up in stroke-like episodes?
How would you measure it? Because
you can measure succinate on MRS of brains, and I don't know if anyone's ever done an MRS with the succinate setting in an acute stroke-like episode. Has anyone here done it? I haven't.
We've tried to with MRS. MRS is magnetic resonance spectroscopy, so you're actually not looking at T2 and T1. You're actually, on relaxation times, you're looking at the actual chemical species. So we've tried that unloading with C13, and we haven't been able to see succinate levels even when we've measured it by mass spec because of sensitivity. But that may be improving all the time. So yes, that would be something to do.
It's something we could definitely do. I think the next person who has a patient with MELAS, could you, if you happen to do an MRI, could you also do MRI of succinate?
We also do see some of these getting into the cerebrospinal fluid, but I guess doing a spinal tap is not something you want to do that often.
More questions?
Mike, thank you. This was really beautiful. Anything known about reductive stress, NADH, NAD ratio? Have you tried to image that in the heart as you look at succinate, as you look at membrane potential?
We haven't looked at that directly. However, what's happening at the same time is that the NADH is very highly reduced, obviously, under these conditions. That's also causing a lot of stuff going on. We think part of the stress is that the electrons have to go somewhere. The electrons on NADH are ending up by reducing fumarate to succinate indirectly, and also through the Q-pool So they're all ending up there. So part of the reductive stress, we would say, is the production of succinate, or it may be a relief valve for those electrons have to go somewhere. But the NADH, the reductive stress itself, what that's doing is an emerging area that we don't really know much about yet.
Yep. And in every graph you showed, and every graph that anybody ever shows in animal studies or human studies, there's always a spread. There are some animals that have minimal infarct, some animals that have terrible responses. Do you have any clue as to what drives this inter-individual difference?
Well, I think it's a good sign because it shows that you haven't fiddled with your results. If you ever see anyone with a human data or mouse data on these things that all bunch closely, you know what happens. It's, yeah. The reason for that variability is that these are extremely difficult procedures to do, even in an inbred strain of young mice. Huge variability because of the way the catheters are going. You can get peripheral blood vessels which will affect it, but that's not much in mice. That's more in humans and some other species, which will affect the size of the stroke. But there's so many variabilities from the size of the stroke, the speed of the reperfusion, other blood flow things going on. So there's so much going on that I expect the variability, and I presume that's the same in patients. You'd expect variability in these things.
Hi, Emanuela Bottani, University of Verona. Fantastic talk.
Thank you.
Maybe it's a very naive question, but have you compared your strategy with antioxidant molecules?
Yeah. So if we give Mito-Q, for example, my personal favorite antioxidant, if you give that before eye injury, then that is quite protective, but you have to load that up beforehand. We're interested in what happened, how quickly you could add that. Our argument would be that the antioxidant is acting downstream of the ROS burst. So it could be that the antioxidant, given in conjunction or a bit later, also was helping, because we still have a lot of cell death there. So we want to see, are there other things we could add to a kind of a multi-drug approach? We could see if we can block this, add antioxidants, add cyclosporin, to see was that able to block permanent transition pore, just to see could we get additive effects. We know, for example, that malonate is additive with cooling, so there's separate mechanisms to try and minimize the cell death there under those conditions. But yeah, all those things would be coming through. Yeah.
That was great, thank you. I think as I see the blockages in the heart and the brain, I think of endothelial dysfunction. And there's been, honestly, we reviewed the literature, there's a handful only of papers reporting endothelial dysfunction in mitochondrial disease patients compared to controls. Hence, the NO-mediated, so nitric oxide-mediated vasodilatation response is altered or modified in a mito disease patient compared to control. So something for you to consider when you're thinking about unblocking the heart attacks and the strokes, that in fact the responses by way of NO-mediated vasodilatation might be different to an antioxidant or any other compound.
Yeah, it could be intriguing to look at. We know that NO interacts with the superoxide, forms peroxynitrite, so that would be a way that the NO availability is modified by ROS production by mitochondria, and that has an endothelial effect. So that's how things like Mito-Q lower blood pressure because they decrease the sequestration of NO as peroxynitrite. But I think having the effect of NO, I presume, is going to be just to increase the blood delivery and the oxygen delivery. So having the two together might improve the outcome.
Yeah, it's just you get a systemic hyperemia in mito patients that you wouldn't see in control. Just something to think about.
Okay, thank you.
Last question over there.
Yeah, very interesting talk. Thank you. Could you use malonate to protect grafts before implantation?
Yeah, we've done that. They're very effective at doing a heart transplant model in mice. We get very good protection by pre-treating the heart during storage because organ transplantation is essentially ischemic reperfusion injury. There's a lot of the early damage upon reperfusion, and that can exacerbate subsequent organ rejection because you're releasing damps and stuff. So yeah, that was protective in mouse models and also in perfused human organs.
Oh, okay. okay, I'm going to allow Niels
One,
to have
okay.
one last question. Microphone for Niels. Oh, you mic'd up?
Yeah, I'm mic'd up, so I can talk. So this was very elegant and I think well supported by experimental data. So my question is, so there are several other dehydrogenases feeding electrons into the Q pool. So will they be blocked during reperfusion? Can some of them even run backwards and contribute to the pathophysiology? Have you looked at that at all? Or?
In other models, we've tended to, in say cardiomyocyte models, where you can look at it in more detail, we don't see other substrates going up, but because the succinate's both oxidized and washed out fairly quickly, what we think happens later on is that we're getting other substrates are coming back in at that stage. Because malonate also is reversible, it washes out as well. So we don't think it's having a long-term effect on that. What's happening in terms of reperfusion, in ischemia with other electron donors being affected. In inflammation, we see things like a dihydroxyorotate, dehydrogenase being involved, but I think that's kind of really just in cancer cell models where its rate is so much higher than it would be in vivo in most tissues.
So, thank you. I think we're going to wrap up now. Thank you, Mike. That was a fantastic lecture, as you can tell from all of those questions that you had, and a great ending to this meeting. But do not run away, because I'm now going to hand over to Amel and Michelangelo for some very exciting announcements.